Hi, Steve Kirsch here. I'm here with Dr. Kevin McKernan and Professor Myron Bridle. We're talking about this plasmid contamination in the vaccines. And I've got a lot of questions. I think you probably have a lot of questions. We're going to go through them with these experts and get your questions answered. And I'm going to try to start with kind of the most serious and the ones that are on most people's minds, which is, you know, the press isn't making a big deal of this. The medical community is not in arms. Nobody's calling for the stoppage of these vaccines because the experts say there's nothing to see here, and so there's nothing to worry about. So let's start off with that, because that's the elephant in the room. Because when I've talked to Kevin and Byram about this, they say that this is very serious. You cannot write this off, and that these so-called experts, that they're saying there's nothing to see here, it's all based on speculation, and this could be extremely, extremely serious. Is that correct, gentlemen? Byram, you want to start?
Sure. Yeah, you're 100% correct, Steve. We don't have, you know, Kevin and the others who have been directly producing these data, right? Kevin made this discovery. Lots of other labs have.
Yo, what's up, Greg? How are you, bro?
I mean, we're lacking the data to definitively state what exactly could be happening biologically, which means since Kevin and his colleagues are the ones who have generated and have the data, these other experts who are being asked, they do not have the data. They have no idea. Yet they can only speculate. What we do know, this is very, 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 very important. Health Canada, right, the equivalent of the FDA, but in my country, Canada, Health Canada has confirmed, and they seem to respond once Kevin reported his findings. They went and looked, and they have confirmed the presence of the SV40 promoter in the bacterial plasmid DNA that Pfizer used to manufacture their shots. Health Canada has also confirmed that it is a bioactive genetic sequence, right, which means that it can do things in the body. And they can't definitively say or rule out the potential for harm, but they have admitted that it's bioactive if it should be contaminating the vials. Now we have data due to the work that Kevin did with Dr. David Speaker, a virologist in Canada, showing that all the vials they looked at for Pfizer in Canada were also contaminated with this bacterial plasmid DNA. So indeed it is there, it is being injected into people, and Health Canada has also confirmed that that was not disclosed to them by Pfizer, and finally they've confirmed that that is against their rules. It breaks their rules. So no, nobody can say
All of the above.
that this can cause no harm. Nobody knows.
Yeah, yes.
Kevin? You agree with
I
what Byron
appreciate
just said?
your position on this, and I'll just one minor correction is I'm not a physician or a doctor, so I just want to let everyone know that my position here is really good. I'm good at studying DNA. It's there. All right, we've got that confirmed all over the world. 6 different labs. We would have it in 12 more labs. We've got a list of labs asking for these PCR kits, and we couldn't supply them because we just didn't have enough of them. So we finally have them manufactured at some scale where we can give them out to more people. But it's there in the vials. The next question is it in patient samples that have been injured? Is it in patient samples that haven't been injured? We need to look at both populations to see what's going on here. So for those not familiar with what this bioactive molecule is, this SV40 enhancer has been published prolifically as being a tool used in gene therapy. So it takes DNA and drags it right to the nucleus in hours. We can also forward your audience other papers suggesting that when you do mammalian transfection like this, a subset of the cells, somewhere around 7 to 10% of the cells, get permanently integrated when you bombard them with linear fragments of DNA like this. So to say that this isn't an issue is really, I think, willful misconduct. You have to take a precautionary principle and say, no, wait a minute, we don't know if this is an issue. We know there's DNA in there that shouldn't be, that was never disclosed and never consented to. We know it's in a transfection vehicle, and we know it has a sequence that drags it to the nucleus. Those 3 things are enough to ring alarm bells to stop. Stop and look to see if there's integration happening. Because if there is, there's a whole host of other complications that we have to begin looking at.
Yeah.
Go ahead.
Yeah, I mean, at a very minimum, the regulators should be warning the public. They should be saying, whoa, we have a major problem here. We're looking into it. You probably don't want to, if you don't need to be vaccinated, don't be vaccinated right now until we can look into this. Isn't that what the regulators should be doing right now?
I'll comment from the perspective of Health Canada. Steve, yes. Again, I can't emphasize this enough. Health Canada has stated in an email, so it's publicly available. They've stated in an email that this is not supposed to be there, and they've confirmed that it's a bioactive molecule. It's not supposed to be there. It breaks the rules. I mean, if they want to maintain any thread of trust in them by the public, they need to immediately put a moratorium on this. They need to, like any other drug that would be found with a contaminant that was not disclosed to them, would be recalled immediately. That is exactly what should happen. So I actually disagree with you. I wouldn't say if there's anybody who doesn't need it, don't take it. I would say Health Canada, the onus is on you now. You admitted this months ago, but you were wishful, or you at least were okay because Kevin's data was reserved to the U.S. It wasn't linked to your regulatory policies. But now Kevin and David Speaker and others have shown that it applies to Canada's vaccine vials. So now the public knows this, and the onus is on you. You have a contaminant now that you have publicly disclosed to everybody, is not supposed to be there, was not revealed to you. It breaks your rules. This is the case, Steve. How many lies do you have to tell to be a liar? One, right? So if Health Canada wants to restore faith, they have to immediately recall this product.
But they haven't. They've done nothing. They've done nothing other than confirm it.
That's right.
I'm really confused on what they're waiting for. The adoption rate of these is so low. Like, what thread are they hanging on to keep them there? There's very minimal risk for them to pull these at this stage when no one's taking, very few people are taking them anyway. I think it's important nonetheless that we push on this because there's children and pregnant women and a host of people that don't know this information that are getting hit with it.
And can I mention one?
Schools banning them.
Yeah, can I mention one more thing, Steve, based on what Kevin said? What's very important is Kevin and David Speaker and several other authors have this preprint that they just put out, which includes this Canadian data. But what also is included in there is very important because it backs up what Kevin was saying. I agree. When I say that there's no hard evidence of harm, I'm talking about those people who are saying there's nothing to see here, right? There is no problem. They have no evidence of that. Like Kevin mentioned, there are lots of theoretical harms, lots of potential mechanisms whereby this bioactive genetic sequence could cause harm. That is in and of itself sufficient. It should be sufficient in science to say we've got to stop, we've got to do the research now to definitively answer these questions. But get this, what they showed in this preprint article is that the batches, the Canadian batches, they were able to link them to adverse event reports, and the batches that were most contaminated were associated with the highest number of adverse events that have been reported. There's a correlation there, right? So in fact, there is some evidence in the hands of Kevin and his co-authors, right, that does suggest harm. It can't be ignored.
Irreparable harm, by the way, right?
Yes, and that correlation was evident with Pfizer. It wasn't evident with Moderna, but we need more data. There obviously could be confounders here, but this is what VAERS is designed for, is hypothesis generation.
Yeah.
When you see signals like this, and there's other data that's not in the paper that is public that people should try to. If you plot those on those charts, they'll reinforce this. Like Philip Buckhalt's lots weren't in this because they weren't from Canada, and those have even more DNA than what we found in Canada and higher adverse events. We also have another lot that's come in, FL0003 from Pfizer, that is off the charts on DNA concentration and off the charts on adverse events. They're the lots that were in Germany. There's a couple other lots floating out there that all sort of reinforce this trend that higher concentrations of DNA are having higher adverse events. So I think as more data rolls in, that chart will begin to reinforce itself on the Pfizer side. I don't know what's going on the Moderna side. There's a lot of other different components to Moderna. They have lower DNA levels, but their adverse events don't seem to be tracking with the DNA concentrations at the moment. But we'll see if that changes when more data rolls in. Again, Moderna doesn't have this SV40 contamination system, so they're kind of on a different playing field right now.
Yeah. So that, but if you're stuck in the middle here in terms of, hey, some experts say this and some experts say this, the data that you have is moving it towards the, hey, folks, this is very serious. It's not moved. The data that we have, the little data that we're getting, suggests that this is much more serious than there's nothing going on here. In other words, if you had to call the odds here, it's like if you were to call the odds about that this is serious or very serious, would you give me 60-40 odds? Would you give me 80-20 odds? What's your feeling if I were to ask you for a number?
Higher, 95.5. Only
95.5.
We've got evidence there being a problem here. It was not disclosed to the regulators. Theirs is on fire, and we now see this contaminant in many vials internationally. So I don't think they should leave them any wiggle room here on what needs to be done. This is a hit the brakes moment.
Okay, Byram.
Yeah. So my comment. So I'm a scientist and a hardcore researcher, right? So when you ask a question like that, I find it more difficult to put numbers on it. I don't disagree with Kevin at all. So the way I would put it, though, as a researcher is you look at what the existing data is, right? There's a research question that's been posed, so I view the question that you put to me as being a research question. And then you say, okay, I look at the existing data, so what would be, and understanding the mechanisms in the existing data, what would I predict? In other words, what would my hypothesis be as I move in forward with this kind of experimentation that Kevin's going to do? My hypothesis would be that it's going to be found to be harmful. Yes.
Chris, what if I were to ask you for odds? Would you? Kevin said 95.5. What are your numbers, Chris?
Well, hi Steve, hi Byram, hey Kevin, good to see everybody.
Chris, good to see you.
Likewise. So I don't know yet because a lot of my research to this point has convinced me that the spike protein itself is toxic, but I differentiate it. I think what I'll call wild type spike protein, because there's a lot of them, right? There's the Omicron spike and the Delta and all that, right? But this obviously probably grotesquely misfolded pseudo-uridine out product that comes from the vaccines has its own toxicity profile. I think we've got good data to say that alone is doing something. And now we've got this plasmid on top of that, so I don't know where to differentiate these effects yet, but I can tell you, just at an FLCCC board retreat, and I get to query Pierre Corey up close. Just talking with all the doctors there who are frontline treating people with vaccine injuries, it's just grotesque, and it's a really wide-ranging soup of conditions that are hitting them.
And these are very long-lasting, by the way. People who got their shots when it first came out, they are having new symptoms. Like I know someone who got shot when the shots rolled out, and she was doing very poorly, then much better, then poorly, then better, and she's been better for a while. And I just got a call last week that she lost use of her legs. And was fine before.
Well, I've been waiting for the all-cause mortality data to die off, because I thought, correlation here. I was thinking it was going to track with some lag, the overall rate of vaccine boostering and uptake, and it just keeps going. So I don't.
Yeah.
These are really tricky signals.
Deceptive.
And I should clarify my point. I wasn't saying 95.5 that it definitely harms people. This DNA. Just that 95.5 that this is a material event in the disclosure process, and that it's going to be legally significant.
Right. Okay. So you. Okay. Great.
Yeah. If I were asked about the legal, yes, I would say 100%.
Okay.
I mean, from what I've seen from Health Canada, it's 100%.
Right. So in other words, I mean, even like best possible case, at a minimum, these regulators should be letting the public know, hey, we may, Houston, we may have a problem here, right? That they should be saying that at a minimum. Now, what they really should be doing is they should be saying, hey, let's take these things off the market, right? Would you all agree with that? That they should be taking them off the market for now until they can investigate the harms?
I mean, that
Yes.
data was obvious years ago, but this is just one more really large nail in the coffin.
Yeah, so when you say years ago, some people said, oh, hey, we knew in 2021 that the SV40, that there was SV40 in the vaccines. Is that, or SV40 promoter sequence, did we know in 2021 that
2021? No, we
No,
didn't.
this was Kevin's discovery.
Yeah, the material they gave to the EMA, and I'm assuming this is what Health Canada has been given as well, annotated everything in the plasmid except the SV40 region. So they were afraid of this. I mean, you don't go about annotating a plasmid with all the detail of the antibiotic resistance gene, the T7 promoter, the spike protein, the cut site they used to linearize it. I mean, they went and annotated this thing with all of these details except the most material piece, which is the SV40 promoter that's active in mammalian cells. So to me, that is they're clearly hiding it.
Okay, so they showed the gene they had the regulators had the SV40 gene sequence, right? The promoter gene.
So they had the entire plasmid sequence.
Okay.
They had the map, and the map you sort of paint on the map what the parts mean. There you go. And what they didn't paint was the SV40 region. Although they gave them the sequence so they can kind of slip it in, but they didn't spell out what it meant.
Okay,
So you see, Steve, this is what Kevin's talking about. See, it shows cut sites, it shows all the bioactive genes that are in here, everything, and you can see all the labels. But they didn't. So they disclosed all kinds of things, but not that SV40 enhancer. And can I just tell you something, just really quickly for your listeners, Steve, because I think this is really important. Listen, I've got the quote. This so your listeners understand. This is what Health Canada, your equivalent of the FDA, said in an email. This is my words. This is what they said. Listen, Health Canada expects sponsors to identify any biologically functional DNA sequences within a plasmid, such as an SV40 enhancer. And this is to be done at the time of submission. And then it says here, that Health Canada says...
Wait a minute. It said that?
Yes. Yep, and then they go on to say the sponsor, which of course is Pfizer, right, did not specifically identify the SV40 sequence. And then they say it was possible for Health Canada to confirm the presence of the enhancer based on the plasmid DNA sequence submitted by Pfizer against the published SV40 enhancer sequence. So again, that's what we just talked about. They submitted the sequence as a data file, right? So they have a computer file sitting there with the sequence. They could see, and then they looked at Pfizer's document after Kevin published his data, and they said, my goodness, Kevin found this, but it isn't listed anywhere on here. But we do have the gene sequence, so we can go back, and Kevin's the expert on this. He can tell you, you can blast that in a publicly available database, a beautiful one that your government offers. And in a few minutes, you can show, bang, 100% match for the SV40 enhancer sequence in the plasmid. That's what they did.
Right. So it's the equivalent
dude, that looks
for
sick,
the lay public. This is the equivalent of a bill in Congress where they have this long bill, it's 500 pages long, and they sneak in a little piece here that says, and now we're going to strip all power from the president or something like that. And they don't call it to anyone's attention. But they could say, yes, it was in the bill that you signed, but they didn't call it out. It's
not as simple as that. As I just mentioned to you, Health Canada themselves has disclosed Pfizer is required to disclose to them any bioactive sequence. So it's worse than that, because it's not just that it got snuck in. It's that it got snuck in, but it's the equivalent of there being a rule. You cannot sneak anything in without making it explicitly clear to us that you inserted that.
Yes.
Okay.
So we have a diagram on the screen here, and this is Kevin's screen, and this is, there's a duplicate of this in Kevin's video testimony that was on the World Council for Health. And so if you want to go through this and explain this with the mouse, Kevin, that'd be awesome.
Just to give you a sense of what they... So this is the plasmid they gave to the EMA. I'm assuming this is what they gave to Health Canada. You can see they've annotated the spike protein here. They've put in this F1 element, this poly-A linker. They even annotated this little 5 base pair sequence known as the EMA 1104I site. That's a restriction enzyme that cuts open the plasmid and make it linear before they run an IVT reaction. They put the ORI in here, they put the CAN in here, genes. What's not in here is this whole region in here has no annotation at all. And this is where, now sorry, but these images are sort of mirror images of one another, so you'll have to see that the neo-CAN gene is over here in green, and here's the SV40 region, which is not here. And what's really critical, they also left out the F1 origin, which makes single-stranded DNA, and I don't know if that's active in mammalian cells, but it's another region that they left out. But the most critical part here is this 72 base pair enhancer. This tandem repeat here is published heavily for use in gene therapy. It drives DNA straight to the nucleus in hours, and David Dean does a lot of work on this. So I think this is the reason they hit it, is that they probably started manufacturing this and realized, oops, we left in a mammalian promoter. That's not good. And that means this plasmid DNA is active in mammalian cells, and it's not just active in the cytoplasm, it goes straight to the nucleus, and that increases integration risk. And since we're dealing with LNPs, this DNA is going to get there uninhibited, and it probably got deleted from their disclosure. Because other words, I don't know why you would annotate something that's 5 bases down here, and even a T7, which is maybe 20 bases up here, and a Kozak consensus sequence. I mean, you're getting down to fine-level annotation of 5 base pair elements. And then you emit this 366 region base. 366 base pair region, that's like probably one of the most functional regions of the plasmid. So to me, it's clear someone hit something based on the details that are in this map.
So it was intentional. It was intentional. Is that what you're?
I think when you put this,
you're speculating. You're speculating, but you speculate.
I want to back up the speculation. People don't go into plasmids by hand anymore and annotate these things. They put them in tools like SnapGene, which is what I did. You download this for free off the internet, plug the sequence in, and it paints all of this stuff on there. So I would have to actively go and delete this stuff from SnapGene and then give it to the FDA.
Yeah, and anyone can go to SnapGene and do this, by the way.
Yeah, anyone. I have something in my Substack that teaches people how to put a DNA sequence in the SnapGene, and they can put this in there and see that it automatically annotates all this stuff. And you'd have to actively go and delete the information to get rid of this stuff. So there's no way in my mind that they just oops forgot about this.
Dude, this dude looks so sick, man.
Painted this, and someone had to actively go and delete it.
And Steve, I want to back that up, so because I hadn't even thought of it from that perspective, Kevin. You're absolutely right. My lab uses SnapGene as well because we make
that looks insane.
virus vectored vaccines all the time. So there's all this cloning work that goes on, and SnapGene is used. And so I can confirm Kevin's right. It would automatically label this. I never thought about that actually. It's a very interesting point. So they would actually have to have removed the default label.
Right.
Maybe a little less.
Comment?
Well,
Yeah, like that.
how they could have left that out. Particularly, Byron, let me be clear about this. That thing you read, they weren't just, that was instructions given before COVID, before this vaccine came along, where they said, for instance,
That's so sick, man.
That's something they put in
Look how sick
Nope,
that is,
nope.
dude.
That's what they even said. That's what they say. That's the important point. Health Canada expects sponsors to identify any biologically functional DNA sequences within a plasmid, such as an SV40 enhancer, at the time of submission. This was in existence prior to that, because of course, Health Canada looks. They deal with gene therapies and everything else. That largely comes from the gene therapy regulations, right? They
I like it.
the rule is supposed to be that they be made aware of any potentially bioactive sequence and any genetic material that's submitted to them to regulate, and if it's not, it breaks the rules. And my understanding was that was supposed to prompt immediate recall.
Well, but what's the chance that Health Canada or anybody else, the EMA, the TGA, doesn't have access to SnapGene? I mean, shouldn't this be minimally what they would do as part of their job?
That's what I've learned from this, Chris. You're absolutely right. It raises for me a huge red flag, because I can't definitively say whether they do or do not do that. But I'll tell you this, Chris. Moving forward, I would certainly like to hope that every regulatory agency will do this in the future. I mean, to me, again, Kevin is the real expert on all this molecular biological work, but it seems to me that this should have been part of the quality control process.
Yeah, so let me clarify something for you, Chris, because the press release specifically called out the SV40 promoter. The press release did. But it's not in the Health Canada regulation that you must disclose the SV40 promoter sequences such as the SV40 promoter. In other words, what they wrote in the press release is a more embellished interpretation of their rules. The rules didn't say you shall always disclose if there's an SV40 promoter sequence in it.
No, but it did. No, but it's bigger than that, Steve. It says they have to disclose any biologically functional DNA sequence.
Correct. Correct.
Which includes SV40.
Right. No. But the point is that the Health Canada regulations on the books do not specifically call out SV40 when they wrote the press release. That's when they added the SV40, because they're not mind readers and being able to predict the future here. They basically just said, you have to let us know about any stuff that's in there that is bioactive.
Yeah, so in other words, the regulators, they're letting Pfizer submit their sequence, and then they're letting them submit their map of the sequence, and then they're just trusting. They're trusting them, having complete faith. To me, I agree with Chris. Where's the policing? Really, we're going to let the big pharma tell us exactly what's in there, and we're never going to question it?
We're the police.
I will show you. Let me show you something that should have shocked them if they had opened up SnapGene. Can you guys see the screen?
Yes.
All right, so this would have stood out to anyone who opened this up in SnapGene, which is, why the hell is there an open reading frame in both directions of the spike protein in Pfizer? Like, that is a very long, unexpected open reading frame. So that's an entire start codon to stop codon of a gene that runs the opposite direction of the spike protein on Pfizer's vaccine. This would have been a major alarm bell if they just loaded the vector into SnapGene and looked at this. I didn't do any work here. SnapGene highlighted this for me. All right, this has been published too in other papers. This isn't some spooky thing that's come up on my computer. Like, people have made note of this, that the Pfizer plasmid, interestingly enough, can be read in the opposite direction without any stop codons for 1,230 amino acids or something. All right, so that's going to be a bioactive molecule. Now, when you blast that, it doesn't hit any human genes. I don't know what it is, but it's not going to be recognized as self. So it has the potential to make a very large open reading frame of non-self material in the plasmid that's being injected into people.
And this
so that would have been well.
This has been known since how long? Since 2021?
Ever since the sequence came out. Let me see. The first sequence that came out was from Andy Fier's lab. They did RNA sequencing, and all they published was they didn't find any vector. All they published was the spike protein region. And I think somebody since then, I have to dig up the right author on this, I'm forgetting their name, had noted that there was an open reading frame in the opposite direction on the spike protein that was uninhibited due to their codon optimization. But everyone wrote it off, being like, well, single-stranded RNA, you'll never have that strand around, don't worry about it. And now we know there's DNA in there, that strand is around. And that's another bioactive molecule that should have stuck out to any regulator who would like put this into SnapGene and open this. They would have said, all right, there's SV40, they didn't tell us about that. There's an F1 origin that makes single-stranded DNA, they didn't tell us about that. And what the hell is this 1,200 amino acid ORF doing here in the opposite direction on spike? That's not part of the virus. The virus doesn't have this. This is a codon optimization artifact that shouldn't be there.
Wait. Is this on the antisense strand?
It is. Yeah, it's on the other strand. So we don't know if it's got a promoter or not, but
would the bacteria have read that and created RNA?
So this is a really good question. Is how do you predict internal ribosomal entry sites? Which is not, there aren't good computer programs to look for irises, internal ribosomal entry sites. This is a place where ribosomes bind down and move. Now, the second question is, will it make RNA off of this? Well, there's an SV40 promoter over here, which is this 72 base pair piece is a bidirectional promoter. It makes RNA both directions off this molecule. So it will make an RNA that goes this long. The question is, will the ribosomes read it? Because we don't know if there's a Kozak consensus sequence anywhere in here. I didn't see one, but I'd ask others to go and look. Because
what's up, Moonraker? How are you, dude?
Elements are hard to bioinformatically predict. But
6 a.m. there or something,
even
isn't
if
it?
you chop all this stuff up and it integrates, you're going to end up getting these
What's up,
little
bro?
open reading frames that are likely to be in frame.
Good, man.
That get
That's
integrated
nice.
and have a short peptide that's non-human that could get
7:30. Oh,
cells,
nice.
Right? It's just something that, when you're doing codon optimization, this is a flag. Like you don't want to have the opposite strand be fully coding because it's more noise in the system.
So it creates risk.
It creates risk, and it's a sign that no one looked at this. Pfizer gave them the sequence. They put it in a file, and that was the end of the story.
Right, and nobody's asking like, why did you do this, and how come, this is there, and why, aren't you doing something to fix this?
Why are these components needed? You can pull up Moderna stuff and see they don't have these things. So you're like, okay, one manufacturer doesn't have as many risks as the other. Well, are these necessary? What's the point?
Yeah. Okay. So let's take this down from the.
So that's interesting. Since Pfizer's been pushed the hardest, right, globally.
Yeah, and the other thing in here to keep in mind, right there, that's the promoter. The AMPR promoter is the promoter that Moderna uses. Pfizer has it too. So all of this stuff here is just all risk, no gain. You don't need it.
Duplication, yeah. I agree. Like my understanding is, correct me if I'm wrong, Kevin, but the ideal plasmids to be using for this are the ones that are stripped down to the minimum bioactive sequence that are needed, right?
No activity mammalian cells, right? Just get rid of anything that's potentially create complications.
Right. Yeah, I've just got a quick question for that sequence that you were just describing, because that's a concern for me as well. Are there any sequence similarities to any known human proteins?
I'll send you the sequence board. I quickly blasted and didn't get any hits that were of interest or made any sense. But I've been told that I shouldn't just rely on NR and blast. There could be other protein databases I should go searching through to see if there's something else that comes up.
Yeah, if you don't mind sending it to me, I'd like to have a look. So, because you're right, because if that gets expressed, that could be like that's something that the immune system will definitely respond to. So it would be very bioactive. And this all links back to, I think, what Chris was saying as well, and you, Steve, with this idea of the timing. With this, especially this new information, I've been swamped with questions from people, like what are the safety implications? And a lot of people have done their research and recognized that this bacterial plasmid DNA can be very long-lasting. Like Kevin said, especially if any of it gets integrated, then it's going to be there for the rest of the cell's life. And to me, it links to what you were talking about adverse events long-term. And I've said to people, and they asked me, I said, I wish I could tell you, but I can't predict what long-term consequences would possibly be because we don't know. We haven't been able to study this. But that's what we have to highlight here, right? These plasmid DNAs, we were told the modified RNAs would be gone quickly, within 36 hours in the body. We know that's not true, but these plasmid DNAs, they can confirm, plasmid DNA, bacterial plasmid DNA, is a very robust molecule. And that, so it's this kind of stuff that could potentially be a mechanistic explanation for the things you were observing, Steve, right? Why things can be happening well after having received shots.
So the question is, why is it
Nice
there?
spot for it. Look at that. That's way better.
Why is it?
Oh my gosh, I think we're approaching
this was the expedient
peak
and we needed to get it out quickly.
stream
and
setup.
Our manufacturing process kind of left it in, and we didn't do anything to
That's
clean
way
it up
better
because
setup.
we knew the
Oh my
regulators
god.
would look. Or like, what is the rationale for all of these things being kind of in there? Because they all create.
I like
risk.
it.
Why are they there? Why do you think they?
I even might move Totobro's thing to the right as well. What if I do that?
Activity. I can't get in people's heads and understand intentions, so I don't know that this is like nefariously put in there. I think when people move very quickly, I suspect Pfizer's vaccine or the plasmid, the idea behind that plasmid was, hey, we could express this in mammalian cells, and that could be very helpful for us to study what type of protein is made. So let's put it in a vector that works in both bacteria and mammalian cells. That way we can shuttle it back and forth, and there's all types of more biochemistry we could do. That makes total sense to me that someone would do that, and then probably graduate into the vaccine. Yeah, if you're a researcher working on this spike protein, you want to put it in a plasmid that has some universality to it, so you can study it in mammalian cells and then amp it up in bacteria.
But that's using human beings as guinea pigs in your experiment, right?
I'm suggesting they were probably they had the interest of maybe putting this in HEK cells or something else to see. Okay, let's express the spike protein in mammalian cells and then mass spec it and see how it oscillates or whatever. Like, you could use the plasmid as a model to study the production of the actual protein,
but not in a vaccine that is given in billions
no, of course,
of doses.
Yeah, they should have removed it, but they probably just ran with the research plasmid and used that as the production plasmid.
Yeah, I concur. I concur with Kevin. Right, we've all been told, we were all assured that no corners were cut. But I agree with Kevin. It makes sense. What he said is exactly exactly how you would design something for the research stage. So to me, this was for the preclinical and translational research phases. And to me, this is probably exactly what Kevin said, an example of the warp speed rate at which these were developed coming out in the fact that they didn't have the time to redesign the plasmid, like Kevin said, for the clinical use.
Right. Now this is
And they
making
probably
sense.
thought they could get rid of it all, right? Oh, we're just going to erase that, so we're going to cover our tracks and no one will know that we used the research plasmid.
Exactly. And so, Kevin, I've got a quick question for you. I think it's very important to Steve, because I just like to get confirmation from Kevin, because I respect his expertise in this as being far greater than mine. Because it was confusing to me. So I'll disclose, I sat on the advisory committee for a graduate student over the past couple of years who was developing an mRNA vaccine for use in poultry, actually. And of course, we advised that after the mRNA was made, so it wasn't the modified RNA, but after the mRNA was made, to get rid of the DNA. And they used DNase, an enzyme that can digest it, and they had no problem getting rid of it. So I was very confused at first, but then I saw a paper recently suggesting that the issue here is with these modified RNAs, that you get all of these odd, like multi-strand, in fact, like DNA and RNA combinations and stuff, and apparently it makes it very difficult to actually get rid of the DNA. Is that true?
It does. In fact, if you look at Moderna's RNA, they have a 100-fold more spike DNA than they have vector DNA. So when their DNase goes to town, it clears out the backbone of the plasmid, but it can't digest the spike DNA as well. And that's presumably because there's a lot of spike RNA that's protecting the DNA from nuclease activity. So when T7 makes RNA and you're putting in these modified nucleotides that alter the melting temperature of the RNA, and they're really sticky, what happens is you get a triple helix. You get an RNA an R loop, which is an RNA DNA hybrid. And some nucleases are good at resolving that and some aren't. They use DNase 1. If you go through just simply through any of these catalog, you would not use that. You would use DNA XT, which is actually designed to get rid of R loops. So there's other nucleases that can be used to clean this up. And I think people should know that this is a very fixable problem. And I think even if they fix it, I don't think they're out of the woods. Well, I just want to emphasize the fact that this moved so quickly that they used the first enzyme you would pick, which is DNase 1. But there's many other enzymes that have been evolved to actually do a better job at solving this problem that they never got to.
Okay, so
I wouldn't be surprised if they could very quickly.
So Kevin, I find it very interesting. I didn't realize that so it's interesting that you've identified other DNases that could be used to more effectively get rid of this stuff. So with that, my understanding is, looking at your preprint article, your very recent one that you just released a few days ago, that it was the first evaluation of Moderna's newest booster. And I'll tell you, so I know, and of course I work closely with David Speaker, who was doing a lot of research. I told him I'm 100% convinced they will fix the problem and got rid of it. But yet it's still there. The DNA contamination is still there. So there's the question then, if, like, for somebody like you, they could have called you up and picked your brain about, okay, you've identified. See, this is what research does. You identify a problem and you come up with potential solutions. So why do you think they wouldn't have got rid of that? Because then they could say, for all this stuff you're raising, they could say moot. It's all moot point, right? It's all the historical shots.
We fixed it.
Yeah, we fixed it.
As long as they have a liability waiver, everything's moot and irrelevant. So there's no motivation for them to fix this, which is why we really need to lean on the regulators to push on this, because they've given them a hall pass. So they're just going to keep claiming it's safe and effective until one of the family members.
In fact, there's risk if they change it, right? There's
Okay, so
risk
this
if they
is
change it, 'cause
interesting,
they'd have
though.
to then do another clinical trial, and they'd be back to square one. So why change it?
But this is the interesting thing. So what you just raised, Kevin, is this legal indemnity that they have, which I was just sharing with Steve the other day, that I read a very recent ruling in Michigan, right? And it was a guy who got badly injured after getting remdesivir, and it was a lot that was known to be contaminated with tiny glass particles, right? It had been recalled, but it was given to him anyways. The hospital hadn't returned their batch at that point, and he launched a lawsuit. It went to court. And Gilead Sciences, who manufactures the remdesivir, was named in the lawsuit. They immediately claimed a legal indemnity, but a court in Michigan just ruled that, it was interesting. They agreed, to Gilead Sciences, they agreed, yes, you have legal indemnity for the approved components of your product, right? That ingredient list.
should not
But you
include
don't
glass
have legal
particles.
indemnity for contaminants. And so would that not apply here to Pfizer?
I think so, because they didn't include, when you look at their ingredient list, there isn't any DNA on it.
Exactly. It's not on the list. I remember we both spoke at the World Council for Health, a few days ago as well, and I showed so we had a Freedom of Information request to confirm, right, what Health Canada actually saw, and Health Canada has now, through a Freedom of Information request, released it, and we also have confirmed definitively that Health Canada, on their approved ingredient list, there's no plasmid DNA, and now they've admitted that with that, in addition, so even if there was that there, still one can argue the SV40, like Kevin said, was not disclosed, so that's not an approved. So even if that were okay, they were okay with some plasmid DNA. They weren't okay with the SV40. So the way I look at it legally, I don't know how Pfizer can possibly escape legal indemnity here.
Well, I think they're going to go, Chris.
One possible way, though, with glass particles, you have the contaminant and you have the harm. They might say, well, there's no harm from this. So can I rewind just a bit? Because we were talking about the possible harm that this might come from this.
Well, you don't know it's harm, that there's harm is the issue.
But they're going to try and evade that. So let me just play devil's advocate here. So they're going to say, they're not replication competent. Even if they were, they're actually only in bacteria, and there's no bacteria in your deltoid, etc. So can you lay out the how would this uptake and do the harm?
Yeah, I think you're right. They're gonna point out it's all broken up. It's within the limits, and the limits are at 10 nanograms per microliter and less than 200 or over 200 bases, and we meet all those criteria. There's some lots that don't meet that criteria, and depending on how you measure them, there's a big debate on that. But let's put that aside. I think what wasn't considered in those regulations is that the DNA be encapsulated in lipid nanoparticles. They were assuming like a 10-minute half-life of DNA in the blood, which I agree with them. If it's just injected, it's going to get eaten apart, and it probably won't have. It'll be an adjuvant, is what they'll say. Is every contaminant they find eventually graduates into an adjuvant category, and so they'll pin it on that as, look, it's doing a little interferon response over here. That could be good for you. But the fact that we know it's in the LMPs means it's materially different than what they evaluated in the past. And we've seen even some FDA guidance from, it's Kilman, I think, is a paper we reference, or Klinman, we reference in a recent preprint that shows if there's any of these bioactive pieces of DNA, they're not, the limit doesn't go to 200 bases, they're worried about 7 bases. Even as small as 7 bases that can integrate and disrupt a gene is a problem. So we really have to focus them on, this isn't about naked DNA. This is about transfection competent DNA going in that's bioactive that could get to the nucleus and create a genotoxic event. And you did no studies on genotoxicity. These were waived. So you can't say that there's no genotoxicity because you gave them a hall pass on genotoxicity. And now we have a mechanism that could induce this. So this needs to be looked at. But I agree with you, Chris. I think they're going to go into exactly what you said, is they're going to say there's no evidence of harm, safe and effective, all these people passed it, and by the way, these nanogram limits we put in place means that we're okay. It's important that people know those nanogram limits were 1,000-fold lower before the Reagan the NCBIA Act. The National Childhood Vaccine Injury Act, they were at 10 picograms before that. They put that in place, and then the nanogram or the DNA contamination guidelines ballooned 1,000-fold in 10 years. There used to be a much lower limit, and the limits that they designed were based on host cell genomic DNA, which at 10 nanograms, you're talking about like 1,200 copies of the human genome. That's not a lot of copies. 10 nanograms of this material is like 200 billion pieces of DNA with active and hydroxyls. This is material that's ready to cause some harm, or at least get ligated into something. So, but I do want to bring up one point that Chris is punching on here, because this is a good devil's advocate position here. I've heard this from others online too, which is, hey, any cell that gets transfected with spikes is going to be dead, right? So why do you care if it's dragging other crap along for the ride? And I've kind of pushed back on that, being like, well, is that true? Is it binary? Is it any cell that gets transfected dies? Because if that's so, how come it shows up in like breast milk and plasma and exosomes and all this other stuff like a year later? I don't know that it's 100% kill rate on transfection. I think there's a population that seems to limp along for a long period of time, and those are the ones we have to worry about for tumors. But is that your understanding, Byron? Because I'm not as dialed in on the immunology front here.
Well, so I can give you one example right off the bat. Immunoprivileged sites would not be susceptible to being killed off, right? So we're talking about places like the testes, ovaries, the eye, the central nervous system, you know, etc. So there's lots of tissues, and this is the interesting thing too. Because I was going to say that then the question becomes, what's the biodistribution of this DNA? And that also links to what you were asking, Chris, because this is interesting. So even what you were saying, Kevin, I agree. If this is free in the blood, it is going to be degraded. But remember, in the blood are phagocytic cells, like the monocytes you were talking about. And there's also macrophages that can be in the blood just before they go into tissues. And there can be dendritic cells. These are what we call the phagocytic cells of our immune system. And these cells they're not really efficient, but they can take up naked bacterial plasmid DNA. But this is the thing. That uptake is dramatically amplified when these things are bound to lipid nanoparticles. And we're talking cation lipids that are making up these lipid nanoparticles. So based on the charge of the plasmid DNA, it can efficiently bind to those cationic lipid nanoparticles. And so this raises two questions that are problems based on what you brought up, Chris. First, a while back you were saying, how do we differentiate between harms due to the spike and harms due to things like the containing plasmid DNA? The easy answer would be, if all the plasmid DNA was free floating in there, right, then you remove the lipid nanoparticles with the modified RNA, you just have that plasmid DNA and you administer that, right, say in an animal model, versus the whole dose, and see what the effect is. But the problem is, like Kevin said, they're married to the lipid nanoparticles, which are married to the modified RNA, which expresses the spike. So we can't parse this out very easily. But my concern then, Kevin, is because they're associated with these lipid nanoparticles, that means they get distributed, like the lipid, you know, to wherever the lipid nanoparticles go, which go to some of these areas.
Yeah, I think if you look at the biodistribution studies of the luciferase mRNA, that's where all the DNA is going. Because I don't think this DNA is on its own. I think it's actually hybridized to the RNA, and it's going into the cell as a DNA-RNA hybrid. Just because the DNA-RNA hybrids are more stable than DNA-DNA hybrids. So there's that thermodynamically, that's what's going to happen. If those two are going to be stuck together, there's a higher concentration of the RNA than the DNA, so all the DNA is going to get pulled out of solution of the RNA, and it's going to get presented to the cell as a DNA-RNA hybrid.
Yeah, and you see, and why that's a concern, Kevin, is because when it's phagocytic cells will often travel, especially if you pick things up to the lymph nodes and so on. So they would be susceptible, I believe, to killing eventually once there's an immune response mounted against the spike protein. But what you're talking about then is that we're talking about any cell being able to take these up. So wherever those lipid nanoparticles go.
You'll notice the regulators spend a lot more time diving into the double-stranded RNA numbers than the DNA numbers. And that's because the double-stranded RNA induces all types of mayhem inside the cell. But I don't know that we have as much information on what happens with DNA-RNA hybrids. I imagine those look pretty ugly to the cell as well. That's a less studied field. So now they're doing something really weird, measuring the double-stranded RNA, which is another area we want to dig into. They're using some ELISA assay to quantitate that, and that should all be done, I think, with next-generation sequencing. Because when we look at this through an RNA sequencing process, we see all types of Watson strand RNA that shouldn't be there under the spike. When you do RNA sequencing, you should get all prick strands, which are sense strands, right? Because the T7 makes one RNA. But when you do RNA sequencing, you don't see that. You see all types of sequence coverage on the wrong strand, which means there's a lot of double-stranded RNA in there that hasn't been accounted for. And the assay they're using is something that, I don't know if anyone's ever validated that on RNA that has this methyl decorated like this. And there's 800 more methyl groups on this RNA. I would imagine that ELISA's probably never seen that before and might be undercounting it. So I think there's a lot of other aspects in here that need to be dug into outside just the DNA contamination.
I agree. And Kevin, I wanted to just highlight one other thing, one thought about what you raised, about how they might try and make the argument, right, that this DNA contamination could potentially serve as an adjuvant. And you're absolutely right in the sense that our immune system, for obvious reasons, right, for fighting off bacterial pathogens, is very good at recognizing bacterial DNA, and it's a potent stimulator of the immune system. But, like, and I'm thinking, for example, of the fact that these shots are being administered to women who are pregnant. One of the things you always want to avoid in somebody who's pregnant is anything that would cause systemic inflammation, because systemic inflammation means it could potentially have an influence on the reproductive tract. And so what's interesting is I would agree with this argument about, maybe it's a good thing because it can serve as an adjuvant if it was like a traditional shot and it stayed in the muscle, or stayed isolated there and to the regional and local lymph nodes, because you want that inflammation. That's the danger signal to draw the attention of the immune system. So you bring in those phagocytic cells to pick up the RNA, make the spike protein, and go and show that spike protein to all the cells in your lymph nodes. That's good. But the problem is when you get this plasmid DNA that can be highly immunostimulatory, distributed again throughout the body with a wide distribution we know is typical of lipid nanoparticles. Now you're talking systemic inflammation, and to me, you can't make the argument when you're promoting systemic inflammation. You can't promote, in my mind, as an immunologist, that is a good adjuvant. To me, an adjuvant that promotes systemic inflammation is a very bad adjuvant, particularly in cases of pregnancy, for example, is a classic one.
Right. Don't eat cheese, but take one of these shots. That's the logic we're given.
Yeah, I mean, it's incredibly, I don't know, what's the, irresponsible for the CDC to tell women to take a shot which causes systemic inflammation.
What else is crazy about this is they're all well aware that the next generation sequencing field just went through this whole transition off of amniocentesis to using non-invasive prenatal testing. So like 3% of the DNA circulating in maternal bloodstream is actually the fetal genome. So you sequence a mother's blood and you can get the entire genome of the kid. All right, that means there's exchange of small DNA between the mother and the child. So this vaccine DNA is going systemic. I would bet it's going into the child, either through the lipid nanoparticle, if it's even naked in the blood. There's probably some exchange there. They no longer do amnios now because they can sequence kids through the mother's bloodstream. You don't have to stick a needle and have a 1 in 500 death rate with an amnio. All right, so there's known communication in between mother and child here, and I would bet on these LNPs getting there.
And you know what, Kevin, I'm so glad you brought that. I had never thought about that as well. But you know what? I actually have taught my students for years about the fact that technically mothers are what we call chimeras. For that very reason, right? A chimera is a combination of two different individuals. We usually think of it like two different species, but same thing with people. So a mother does have a tiny genetic component of their baby that stays with them. I never thought about it, though, from the reverse direction. You're right. So this is why it's important to get scientists together discussing these things, because I totally agree with that concern. That's a major concern.
So if we had a CDC scientist on this call who would defend that, yes, it was the right thing to do to give this vaccine to pregnant women, what argument could they possibly use in light of what you just said to defend?
The only thing that I can think of,
because we're never going to have this discussion, right? Because the CDC scientist is never going to allow themselves to be questioned, because that's not what they do. They censor us. They do not answer any of our questions. If they were here, how could they respond to what you just said?
Yeah, just really quickly, because I put thoughts into the minds of these theoretical, hypothetical people who would potentially speak to us. But what I do know is, their job is to take a balance, right? And you start piling up the benefits on one side and the risks on the other, and their job is supposed to be, if the risks outweigh the benefits, you don't move forward, and vice versa. So to me then, what they do is the risks just keep piling up massively over time. So to me, if I'm to hypothesize what they would do, they're going to try and pile as much as they can on the benefit side. They're going to, but the problem is, they're going to argue this was super dangerous, but that's sort of gone out with work that's showing the infection fatality rate is much lower than was anticipated, et cetera. But you know what I mean? That's what they're going to try and do, Steve. They have to.
Yeah.
If they're going to counter this, they're going to have to get that pile on the benefit side as high as they can to outweigh the ever-mounting risks.
Yeah, and by the way, I just published on my Substack a study of a VA paper. It was published in JAMA, and it basically showed that there was no hospitalization difference if you were hospitalized for COVID versus if you're hospitalized for influenza. The vaccination breakdown for both vaccines was identical in the two groups, which means neither vaccines work to prevent hospitalization. And as Byram, as you and I discussed, if there's no hospitalization benefit, there is nothing in biology that shows that you can have a mortality benefit. So if your hospitalization benefit is zero, your mortality benefit is zero as well, because it has to be less than the hospitalization benefit. There are no exceptions in biology that you were aware of.
I mean, the deeper issue here as well is the CDC conflicted, right? Do they have a motivation to actually push vaccines? I mean, certainly there's a lot of other
Clearly they
run
are.
through there. And don't they have a separate slush fund, the Pharma can fund, that is involved in funding like shots heard around the world? So that, and that I think Bill and Melinda Gates can pump money into. It's not really under their budget, but they're under control of it or under influence of it. So
No,
they do have inside funds coming in. So I don't know that we're going to get honest answers from conflicted organizations.
No, we won't.
You go to the FDA, they have $400 million in royalty from Moderna. They're not going to give us an honest answer. If we go to the FDA, they've got issues in that the PDUFA Act funds half of their budget. They're getting directly funded by the companies they regulate. So none of those are going to come to the table and give us an honest answer. They're going to come and say safe and effective, and the virus is really dangerous.
And just to confirm for your Canadian listeners, Steve, it's very interesting. Health Canada historically, was primarily funded by taxpayers, and I learned during this declared pandemic that had completely switched. So prior to some years prior to this declared pandemic, Health Canada cried foul. They were looking at, and they were so jealous of your regulatory agency, right? And they said to our government, look, they have way bigger operating budgets than we do, proportionately even. So that because they're getting all this money from the big pharma, you're paying all these, they can charge them all these huge fees to review their submissions. And so Health Canada, prior to this, switched over to your system, so that the majority of the operating budget for Health Canada also comes from their review of big pharma. And I've actually looked at their list. It reads like an advertisement pretty much for a service industry, right? Where first submission to Health Canada is a freebie. It's a freebie, right? And how often are you going to reject the freebie if you want the paying business, right? Etc.
That's the free trial.
Yeah, but I'm telling you. So what I can confirm to you is Health Canada gets the vast majority of their operating funds from the very pharmaceutical companies that they are asked to regulate, just like yours. So I agree 100% with Kevin. I believe that this is a global problem, the one that he just raised.
Yeah, and I can tell you, I've talked to a former CDC employee, and they told me that the directive was a shot in every arm. That was their priority. It wasn't about safety. It was about get the vaccine distributed. It was all about the marketing and convincing of people to take the shots. It was not about their mission was never protect the public, safety. It was get these shots in every arm. So do we have, in your opinion, your expert opinion, do we have an adulterated vaccine here? I talked to Dr. Malone. He said, what we have here is an adulterated vaccine. Agree or disagree? Kevin?
I mean, I'm new to this field, but from what I've read very recently, it sounds like it fits that category.
Chris?
Oh, absolutely it does. And I would say because of all the excellent work being done by everybody on this call and the things that I've been seeing, they're starting to lose badly in this particular story. We're getting close to this part of the story, which is called common knowledge, where everybody knows that everybody knows something, right? And so this is when the story can turn very quickly. And just this morning I had a flood trying to keep up, Steve, with what you tweeted out and other pieces coming out. There's that New Zealand Minute MP saying that they had just done a private statistical analysis and discovered that in one location during one day, all 30 people who were vaccinated at this one location in New Zealand, all 30 were dead.
And no news coverage of that, right, Chris?
No, but it's making the circuit out there. And so this is the kind of, we're talking here, gentlemen, about a very comprehensive set of failures that are very difficult to explain individually, but collectively, how did they not put this into, this flu reading thing? How did they not understand this? How did they not, it's just, there's so many collective failures, and we're talking about the reproductive virus. I got a strike on YouTube because in 2021 I was trolling around on some Facebook groups and women were talking about all of their menstrual irregularities, right? Just data. So I put that up there and got an instant strike on YouTube for that, and now we know, of course that's a thing. They can't have missed that in the 60-day post-marketing surveillance study in Pfizer. You can't have missed it, and it's not in there. They talked 1,223 times, but we vaccinated millions of people. Some people die, just random. That's true. They talked about little myocarditis, but they didn't mention the menstrual irregularities. They can't have missed it. It's too strong of a signal. So that was willful for sure, and so we have willful act after willful act after willful act. But hearing that Canada, for the health authority up there, basically be saying, you didn't tell us about SV40. This is kind of an exciting moment because that feels like CYA time. Somebody's backing up and saying, uh-oh.
Got a problem.
I totally agree, and I'm wondering if the Health Canada in some way trying to get themselves off the hook by sort of admitting this up front, and then it was the U.S. that, in the U.S. where all the contamination was discovered. I don't know, but you're right, Chris. So in short, to your question, Steve, yes, this is an adulterated vaccine. There's no question. And Chris, I also got slammed for the bringing up the potential about these menstrual issues, right? And that was remarkable. To me, that's a great example because way after the fact, finally in the U.S., right, your CDC said, okay, let's fund some studies to see if this actually happens. So people didn't want that, but that's a classic example of, yes, the public rollout. The people in the public rollout became the guinea pigs. And then, yeah, you're right. There's been the multiple peer-reviewed published studies now showing definitively that was the case. I agree with you. They had to have seen it before. And that's my whole point, that this stuff keeps accumulating so much. All these risks, all these harms, all of these misdeeds or mistakes, whichever one they are, because if it's the latter, then it shows that our regulatory agencies are completely incompetent and incapable of properly regulating these types of products. Whichever way you look at it, all of us on here have been accused of being anti-vaxxers. I mean, my goodness, I'm a vaccinologist. I still continue to publish vaccine papers. But the amazing thing is, what people have to start recognizing, right, those people who accuse us of disseminating misinformation, all of us have actually been the ones fighting to keep be able to maintain people's faith in vaccines. That's why I've spoken up, Steve, is to protect my field. And now we're seeing all over the place. Pfizer, Moderna are crying foul now because nobody will take these, their shots anymore. The uptake has been so low, and they're losing their billions of profit. And now, but worse, we're seeing uptake of all the other vaccines. So I just before we got on this call, I was just finishing reading. You might be interested in looking at this, Steve. A CDC study, right? So from your country, they were looking at pregnant women, and the CDC is very upset because their cutting-edge data right now show that uptake of other vaccines by pregnant women is plummeting. And they believe it's because it was very interesting. They only got one expert to say, I think, what they wanted to, but they found one expert who said that they believe the reason is, so everybody admitted that it's because a vaccine hesitancy is skyrocketing. And then one person said, oh, that's because of the spread of disinformation. So all of us here, plus all of our colleagues and everything else, I think maybe they have to start waking up and recognizing that when you have a product, a good product, as I say, will always sell itself. And when you have a product that is as crappy as this, and there's been all these problems, and they're still emerging, and they will continue to emerge for who knows how many years to come, you have to start admitting that the misinformation gurus are the ones who have destroyed the field of vaccinology, and they're the ones responsible for the skyrocketing vaccine hesitancy. And I say to anybody who's vaccine hesitant right now, you deserve to be and you should be. And the onus now is on our regulatory agencies to earn our trust back. And you know how they can do it? By being very transparent and honest in all of their regulatory processes moving forward, and showing us very transparent, very well executed, extremely well designed scientific studies to back up their products. That's what I say.
I'll tell you one more thing they need to do. You admit where you went wrong, you explain how you went wrong, and you explain the steps you've taken so that you don't do that again.
Right. And also, you know, the very first thing that Health Canada should have done is they should have hauled Pfizer in and said, how did the SV40 promoter get deleted from those charts that you gave us? How do you explain that? You know, this is like the first move is just to say, hey, how did this happen? And nobody's asking that question. Nobody's asking the question, right? Congress isn't asking it. Health Canada's not asking it. The New York Times isn't asking it. Nobody's asking that question. I'd love to ask Pfizer that question, but they never return my calls for some reason.
Steve, did you see the interview with Freeman in the FDA?
Yes, I did, and
Okay,
I talked with
so
Joe after I saw that. I think that was it on Dr. Drew that they aired that, right? And he should.
Yes, yeah. So an interesting detail on that is that they only consider the adverse events they saw in process 1 as being things that they will then correlate with process 2 to adverse events, right? So if they had a different process 1 that didn't have endotoxin or plasmid in it, and they get some set of symptoms, maybe for some reason menstruation doesn't show up in there, I doubt it though, I bet it did. They then move forward with process 2, get a host more adverse events, but they can write them all off saying, well, we didn't see him in the first process, so we're not going to count him in the second.
Correct.
That's a major goal. They have to get rid of that. That's a disaster.
Correct. Totally agree. And we're just to give people context, I think we're talking about this. There was an episode of Dr. Drew. Joe Freeman was on Dr. Drew. He revealed that he had a recording of a conversation that his team did with the FDA. His team, the short story is his team he spoke with his team after the call, and the team was absolutely appalled at how the FDA does science. Is that fair? Is that a fair recap?
Yes. Yeah, they were not, they were breaking all their own rules. It seemed as if it was obvious that you have to correlate adverse events in a certain way, and they were basically saying, we're not going to do it the obvious way, we're going to do some type of observational thing that we can fudge later. It's worth watching. It was just a train wreck.
Yeah. Yes, it was definitely highly recommended. If you want to understand how the FDA works, watch that Dr. Drew episode with Joe Freeman.
That's insane to me. That's like saying, you know, we dropped somebody off a stepladder on the moon, and we saw a certain number of injuries, and those are the only ones we look for when we drop people off the stepladders on the Earth. That's totally, you know, it's... Yeah.
But they justify it, Chris. The way they justify it is they say that, look, you know, the first process 1 was done in a randomized trial, so we could identify what the real signals were from the fake signals. So when you see process 2, since we didn't have a randomized trial, you really can't assess whether those things were real signals or not because we don't have a control group. That's essentially
Operation Warp Science, right?
Yes, warp science. Yes,
And,
yes.
and also, Kevin or Steve, remember that's after they've removed people from the study like Maddie DeGay because they were outliers and didn't meet the
Right.
full requirements for inclusion in the safety data, right?
Right, and this reanalysis paper that was published in this journal, peer-reviewed journal recently, where they reanalyzed this is a bunch of people associated, Dr. Naomi Wolf's team, and they basically found that the same thing I did, and I think Chris realized this as well, is that there were way too few deaths reported from the 22,000 in each cohort and they pointed out the same thing I did, is if these were truly representative real people, they would have died at a much higher rate, like 5 times higher rate than they actually.
The baseline mortality rate was wrong?
Died. Yeah, based on normal mortality rates. And so they said, oh yeah, about 80% of the people who died were lost to follow-up.
And also, did the paper also disclose, Steve, there was also something wrong with the timing as well? That there was this odd, non-random clustering of deaths as well, because like you said, you'd expect a certain natural mortality, and with natural mortality, you also expect a certain amount of randomness. But there was a lack of randomness, is my understanding as well, in the reported deaths.
Yeah, and well, it was like 3.7 times higher cardiac events in the treatment group versus the placebo group, as one of the things that they discovered. For example.
Steve, so I'll tell you a quick story directly related to that. Myself and several Canadian colleagues found that as well, and we put together this manuscript, right? I still have the manuscript. I tried publishing this a long time ago. We made those observations. We made the observations about the red flag with the cardiac stuff, because where they put all this data, right? They kind of hid it in their supplementary data, right? And very few people, like when you talk with physicians, the average physician, when they go to quote a paper, they pull up the paper, they look at the abstract, they might look at the concluding paragraph, that's it. They don't look at supplementary data. We looked there, and yeah, you're absolutely right, there were more cardiac deaths. And there was no all-cause mortality benefit whatsoever, right?
Never mentioned in the abstract.
Yeah, exactly. So we published a paper outlining all of these anomalies, and so I was so glad to see this paper recently published. It was also frustrating because we could not get one single journal to send basically the same findings, right? A year and a half ago, we could not get the same findings to go out to any reviewer. No journal would send it out for review. It was considered far too controversial. But to me, this is showing, right, things are shifting, because things that were not allowed to be published, even reviewed for potential publishing a year and a half ago, now are. And I think that's because the scientific journals are also recognizing if they want to maintain any sense of integrity, right, in the eyes of scientists who are looking to publish, they better start allowing the real data to come out.
Well, Steve, what are the odds that these journal publishers know somebody who's been harmed or killed?
Oh, gotta be super high. There's no way. Pretty much everybody is and maybe not everybody, but it is very high.
And the Pfizer advertising dollars are probably plummeting. I mean, I'm in Massachusetts and I'm hearing of layoffs, so that means the advertisement when we launched the solid system, the sequencer, back then in like 2008, it was like $10,000 a page for a full page ad in Science, right? These guys have tons of pages in those magazines all year round, so there's a lot of money coming in from the advertisers into these journals that I'm sure is starting to be thinned out a little bit with the stock compression going on with both of the companies.
Yeah, Kevin makes a good point, Steve. I also just read of hundreds of people being laid off at Pfizer. And I wonder what those people think, having supported Pfizer so much over the past few years and their success and everything. It might be interesting if you could round up some of these Pfizer employees who are being let go now. They might potentially have some interesting stories to tell.
Yeah, hey, I'm here. There's a contact me link on my Twitter, on my Twitter I guess it's X now, my pinned tweet on X, and people can contact me that way. But speaking of the press and stories, so let's start with FDA, CDC, WHO, EMA, Health Canada. Have any of these health authorities contacted any of you guys about this? Especially Kevin, because Kevin was the one who spotted it initially. Have any regulatory authority ever contacted you and say, hey Kevin, we'd like to have a briefing? And no.
No, it's been a flurry of fact checkers calling with demands that I answer to them in 48 hours or 24 hours. And here are all the accusations they have of why this work is wrong. There's even one group funded by the Wellcome Trust that wrote a whole article on why DNA gets degraded so quickly inside of the body that it never gets into the cell. So they completely just omitted the fact that, hey, these could be in the LNPs, and the whole thesis of their argument of the article was gone. And the article is really long. It's like it took this person more time to write the article than to go run the DNA experiment that we did to prove it wrong. So, and then she's funded by Wellcome Trust. They have more DNAs than I could ever dream of, right? So,
I was going to say, you're fortunate, Kevin, if you were given 24 to 48 hours. I was often given 3 hours. I would often receive an email at 1 in the afternoon, and the deadline was at 5.
Yes.
Yeah, and you probably learned this too, that when you do, you provide them with robust answers and lots of robust primary scientific data. Miraculously, those fact checks never get published.
No, it never gets into it's the same thing has happened to me, Byron. What a coincidence.
I fact-checked the fact-checkers in 3 separate ones. I did Reuters, AP, and Politico. And every one of these fact-checkers, when you dig down, they were formerly a journalism student at Brown. There's no science. Like, none of them have any science background whatsoever.
No, never.
Yeah, some of them have links to I mean, I know Reuters, I think they share a board member with Pfizer, right? I think the CEO of Reuters is on the board of Pfizer. And then some of the fact checkers, you dig into their funding, and it's, the one was, I think, Health Feedback, who used to be like Climate Feedback. So they were in the climate change grift and moved into this. So they're, never take health advice from a Malthusian, right? It's a bad idea. But
Yes. Sadly though, just to share with you guys, because I've been called as an expert witness in quite a number of court cases that are related to COVID-19, and sadly, those fact checks come up prolifically in court. It's sad how much influence they have on the court process.
Oh yeah.
One of the biggest early ones from AP was by Duque, Beatrice Duque, right? Formerly fashion editor at Teen Vogue, right?
There you go.
That was her
Totally qualified. Totally qualified.
Yeah.
So
I've taken the habit of just post the stuff online and answer it before they have a chance to even read your response. That way, everyone sees the transparency. Here's the email. Here's my response.
Here's what I sent.
No one can answer it. You can't hide anything.
So let me just summarize. In the entire world, not a single health authority from anywhere in the entire world has contacted any of you guys to ask for more information, to discuss this, explain it, whatever. We want to hear your point of view.
No, I even brought it to the FDA and presented it with the 4 minutes they gave me at the VRBAC meeting back in June, and they immediately ignored it and moved on to the XBB 1.5, fear porn they had to perform.
Yes, well, the same thing happens to all of us who present at FDA meetings. It is like talking into a black hole. The only advantage is that the public watches it, and so you can communicate with the public, but it appears that none of the members of any of these committees is ever listening to anything that is said by the public at any of these public speaking opportunities at their meetings.
But they tell me here in Canada all the time, the FDA does, that they use that to tell me that they've done lots of public consultation.
Yeah,
that seems like a very good thing to me.
Yes, and you couldn't tell it from any of the questions or any of the remarks happening after any of these public sessions, because nothing that is ever said in any of the public sessions is ever integrated into a question or a discussion or even a remark from any of the participants in any of the panels. I have never seen an exception to that rule. Okay, so let's talk about the press, though, because I asked you, has anyone from any of the public health authorities talked to you guys and reached out to you? Surely, the New York Times, the Washington Post, CNN, and all the mainstream media, these guys must be hammering you, Kevin. Your phone must be ringing off the hook because they want to talk to you about this story and the significance of what you found.
No, I've been getting the alternative media, yes. So, whether it's Rebel News or Epic Times or, Jan picked it up, Peter McCullough picked this up. You have on your vaccine network, the safety research group that you have, Steve. But no, I'm not seeing anything from any of the the legacy dinosaur media.
Chris or Byram? Any contact?
So, yeah. By the way, I always struggled with that term, Kevin. So I've actually switched it now. I've got my own term I use. I call it integrity media. So yeah, I've been contacted by some integrity media, but no others. And again, I just want to highlight for your listeners just how egregious
Who are you
this is.
contacted by?
We have state-funded media in our country as well. It's known as the Canadian Broadcasting Corporation. But this is what amazes me, because maybe you could understand in the United States why they might not, but there's no excuse in Canada at this moment, because like I said, I can't emphasize this enough. 2 days ago it was revealed, Health Canada themselves saying that Pfizer duped them, right? I mean, that's exactly what they said. Pfizer duped us.
Should be news.
You would think that your national, major tax-funded news agency would want to make sure every Canadian knew, because guess what? If Health Canada was duped, then every Canadian was duped, right? They have not mentioned it at all, and they have had several days now to do so. To me, it's absolutely, egregious, and I mean, we're lost.
Parliament? Was it Andrew Brigham's work in Parliament? Did you see his talk on Thursday or Friday? He brought up all the excess mortality. It was empty. It was a completely vacant chamber.
Yeah. Anytime any MP does that, right? Anywhere in the world, it seems they're talking to an empty Parliament. But hey, in Canada, I mean, I don't know if you saw. I mean, if anybody talks about that in Parliament, it'll be empty. But I don't know if you saw, but our entire Parliament gave a genuine Nazi a standing ovation a few weeks ago. You know?
I noticed that. I saw that. But
Yeah.
They were all present to give him a standing ovation. My goodness. And the poor truckers for the Freedom Convoy were literally labeled as terrorists. And now we got the Hamas going on. And I don't know if you guys saw as well, but literally an email was found circulating through our Canadian Broadcasting Corporation saying you cannot call Hamas terrorists, but they could call our truckers terrorists. Canada's lost, folks, I'm telling you. You have the best chance in the U.S. right now of seeing us out of this complete and utter disaster.
Who was the standing ovation for?
I forget his name, but our whole Parliament hosted Zelensky, right, from the Ukraine, and and they introduced a fellow who and he I forget his name, but he's so what happened is interesting, the history, because they switched their names. The name of his unit, it was a Nazi unit for the SS, right, German SS. They switched their name at the end of the war to the Ukrainian division to hide the fact that they were this Nazi division. And so every parliamentarian stood up and gave him a standing ovation in our Parliament. Meanwhile, history shows that the regiment that he served with, not only was it a Nazi regiment, but in a couple of different countries actually fought and killed Canadian soldiers.
Yep, the actual Nazi. That was Yaroslav Puka, right? He was it was a little bit embarrassing on that whole thing. Hey, Steve, I can answer your question. In the 3+ years now that I've been doing this COVID coverage, I've never been contacted by legacy media once.
Wow.
Not once.
How about by fact checkers?
They come along from time to time, and I've had hit pieces, of course.
Yeah. Okay. All right. Let's talk about these test kits, Kevin. Because the first thing that after putting a halt on the vaccine, the first thing that these regulators should be doing after saying, look, okay, put the brakes on, no more vaccines until we investigate. The next thing they should be doing is investigating and finding out what the heck is going on. But they're not doing any investigations at all, right? But you are. You, Kevin McKernan, private U.S. citizen Kevin McKernan, is basically offering test kits. You had 500 test kits for pathologists, there have already been 6 labs that have been ordering your test kits, and these test
we
kits are
actually have more if we need to scale it up. But basically, we were supplying people with our own stocks, and we bled those out after all the demand showed up, giving them around to labs. And they're mostly testing vaccines just to see if there's a vaccine problem. But now that everyone sees that's real, a larger demand has been coming in for looking at the blood supply, looking at monocytes, looking at sperm, looking at all types of tissues. So we're just in the process now of trying to scale that up so we can get test kits out to people with CLIA labs and can do human subject research. That's where our limits stop. We used to have a CLIA license, we don't anymore, so we can't do human subject research here. But there's plenty of labs out there that can. And there's a lot of patients knocking on our door that we're trying to push that direction to people who are CAP accredited and CLIA accredited to run testing on biopsies, if you will. But yeah, I'm not holding my breath that the government's going to run and do this. I mean, this is like them investigating. Right. I mean,
this is
the
our
NIH
job. This is our job. The public's job is to protect the public. It's not the CDC's job. The CDC's job is to inject you. It's the public's. It's our individual job to go and find out how serious this is by testing blood. And so this is a simple blood test. You just get a blood draw, and you test the monocytes in the blood. And also,
we should look at more than just blood, but that's
right.
That's
no, I know. But that's the easiest. That's the simplest and easiest.
We should look at saliva and sperm and other things that don't require a needle. So
Absolutely.
there's other non-invasive ways that may show up, but we don't know yet.
Right.
Yeah, I was going to say, you're right, Steve. What you've recognized basically is the new medical principle that we have adopted, right? Which is the inverted precautionary principle. That's the formal name. And also, I would point out that
the inverted precautionary principle.
Yeah, Kevin has raised another good point. Again, that I agree with 100%, and I never really thought about it from a policy perspective, right? This is yet, I mean, why they haven't already, because of the demonstration of free-floating spike protein, the long-term, the long-lasting survival of the modified RNAs, etc. This adds another kink to it. Right now we have contaminated plasmid DNA, including with the Pfizer shots component that was never disclosed, definitely is not supposed to be there. So like you said, clearly these are adulterated. There needs to be a ban on using blood from people who have received these shots, right, from our blood suppliers. Because, and to me, that again is very simple research. If they would just acknowledge it, right? What we need to do is we need to get those who provide blood, services for hospitals and surgeries, etc., right? Transfusions and so on. We need them to look at, do it like they do for anything else where the blood supply might be contaminated. Yeah, exactly. Exactly. Just screen. So as I say, screen for the vaccines, the components thereof, which would now include this plasmid DNA, and as I call it, the derivative or derivatives. In this case, because the spike protein isn't in there, I call that a derivative, right? It gets produced. It's like a prodrug, right? It gets produced in the body after it goes in. So that needs to be included. Spike protein, lipid nanoparticles, modified RNA, and now add to the list bacterial plasmid DNA. And you monitor it in the blood over time. And just like anything else, you determine what's the washout period. Now, the scary thing for me is that washout period might be very long, but we need to determine it.
Like how long might it be, Byron?
I'm not. The research. I'll say again, the research is very easy and simple to do and inexpensive. Let's do the research, and then I'll be able to tell you.
Well, we have all that in the Cell paper. The terminal centers had detectable mRNA 60 days post. That's as far out as it looks.
Yeah, but again, they'll argue that that's germinal centers and not the blood, right? As long as it's sequestered in other tissues.
28 days out in plasma, all right, and then the spike protein lasts even longer. Didn't Patterson find it out like past 200 days or something?
Like
blood?
Over a year, I believe.
Yeah.
I know. I think it could be pretty scary. Yeah, I guess my point is that washout period might make it. If you imagine if you start proposing a washout period that could become hundreds of days for a blood supply where the vast majority of your population, right, has got these shots. That would be devastating to the medical industry. But we have to start having these hard conversations, right? The people,
Yeah,
again,
and
it's about informed consent.
doing the research, right?
It's about informed consent. I mean, if somebody's happy to receive blood that's contaminated with bacterial plasmid spike or whatever, fine. But at least allow people to know.
Right. So I wanted to ask about this quantitative PCR to detect DNA, but fluorometry to measure RNA, that manufacturers had managed to mislead the regulators regarding the presence of DNA in the vials. Do you think that is deliberate, Kevin? You know what I'm talking about, right? Do you think it's deliberate?
Absolutely. Yeah, anyone who uses these tools knows that this occurs. And so for the audience who's not familiar with this, if you go through the EMA's documents that Pfizer disclosed, they use some technologies to measure the RNA, because they're held to an RNA-DNA ratio in the EMA, which is different than the FDA. The FDA just has a total amount of nanograms of DNA. But the EMA has a ratio. And so they measure the RNA using a technique using UV spec or fluorometry, which vastly elevates the amount of RNA you have. And then they use quantitative PCR, which only measures specific pieces of DNA. It's very, very specific, and you get log scales less DNA using this. So by being able to cherry pick these different tools, you can present the regulators whatever you want. And this is what we tried to display in our most recent paper, is that we showed, hey, here's the quantitative PCR number, they're under the limit, and if you put them on the fluorometer that Pfizer was using, we don't know the exact instrument they're using, but they use ribogreen, which is a dye that we're really familiar with. If you put it on that, it's 100-fold higher.
Right.
So if you want to basically choose your own adventure through the regulations, let them just pick different tools. Now, the interesting thing about this is if you had to design quantitative PCR to measure the amount of DNA, you have the primers you need to measure the RNA with RT-PCR, yet they didn't do that. And that would be the more logical thing to do, is to measure the RNA and the DNA with the same damn tools so you're normalized. You can measure the RNA with qPCR, you can measure with RT-qPCR, you can measure the DNA with qPCR. Yes, the RT-PCR gives you RNA and DNA together, and the qPCR gives you only DNA. That's all you need to sort this problem out. And what they did instead is they said, we don't like that answer, so we're going to inflate the RNA with the fluorometer and then sneak in this qPCR for the DNA. That way there's a huge spread and we pass the test.
Correct. So the point is that they gamed the whole way that they tested in order to pass the test, so they could say, oh, look, it's de minimis.
Yeah. And that's happening, I think, across the board. That they're approaching the regulators with, okay, you have a limit here. We're going to find a test that gives us the lowest answer possible, and then give you the data. Now, the second thing to know is when they give data to a regulator, the regulators just look at it and go, okay, did you pay your user fee? Okay, we believe the data. Give me the user fee. There's no actual. They don't actually test it. They just say, okay, you have this on paper. We'll believe you.
Thank you very much. And
So you know what, Steve?
You're making me very mad, and for the same reason why Byron brought it up. I'm a scientist. I care, and this is just such a perversion, right? But I was getting really annoyed when we lost the ability to find herd immunity and suddenly vaccines. We didn't know what those meant anymore, and we couldn't distinguish between a case and an infection. Like every step of the way, this thing I love, which is science, which is based on a shared understanding. Reality's hard enough without making it harder. And they're just taking all the stuff that we have. This is captured territory that we just gave up, right? The ability to understand things in common because we use standards and references that's captured territory. Sorry. That's my editorial development.
And I wanted to overlay something on this. What Kevin, I wish we could take that clip of Kevin, just answer your question there, Steve, and show that in every court case related to COVID-19 as an example of this is what a real expert looks like. Why? Because what happens is, I've observed this so much in court, and it drives me absolutely stark raving mad. Because the whole thing about court cases, right, is who is the expert? How do you determine who the expert is? What's their limitations on the expert, right? Are we going to qualify this person as an expert? And the courts, right, being laypersons, they just see something. They see the EMA data, like you were just asking about, Steve, right? Like the Kevin was asking. They said, well, look, the EMA is showing that this is just fine, right? But the real and then you have the fact checkers and everybody else who then keep citing that, quoting that. But then you have Kevin McKernan, right, who can break down the methodology and explain exactly why those data are not trustworthy, right? And it takes that out of the realm of we have to stop in our court saying, oh, the EMA said this, right? Who is the EMA? Who exactly? And how exactly? And that's what I keep trying to get to people. Your real experts are the ones who don't just read the abstracts and the concluding paragraph. They're the ones who actually read the materials and methods section, which is dry and boring and difficult to go through, but because they understand the methods, and then they are the ones who can then assess based on if the appropriate methods are used, the data can then be trustworthy, and then you still have to evaluate the raw data, not the interpreted version. But like Kevin just pointed out, if you're using the wrong methods, the data are wrong, which means the conclusions are wrong, which means the people who are quoting the final line in the abstract are wrong. Honestly, if our courts work this way, I would love that, Kevin. We could just package that up, and before every court case starts, show that what you just did with Steve there, right to the judges. This is how you identify a real expert in court.
Yes, if only.
Happy to help. Appreciate the compliments. But this is what I do every day, so, and I think anyone else who's in genomics knows this. Like, if you want to get a really elevated number for a manuscript, use UV spec and a fluorometer. If you want to get a really conservative estimate, use qPCR. So it's these are.
And
this
if you
is
want
not
to
rocket
pass the
science.
test, you mix both.
Yes. Yeah. Exactly.
If you work for a drug company, you need to use both. One for this and the other one for that.
But you're right, Kevin.
For both, fine. If they use qPCR for
Chad
both,
Toad.
fine.
I was
At
literally
least
just
yeah,
thinking
stick
about
it
your Chad
Keep it
photo
consistent.
with Anna
Exactly.
Kremling. I'm
You know
not
what?
even
I just
joking,
learned
bro.
after it's
I was
been
literally
so
just
long
thinking,
since
like, I don't think about that. That isn't what goes through my head all day.
You know, since I
Not
went
gay,
public with the
but
biodistribution
I was literally
data
just
for
thinking about that. What's up,
Pfizer's,
man? I'm gonna weigh myself right
shots.
now.
But just brought to my attention, Kevin, a fact check that happened way back then, where they were
How are you, man?
arguing,
You're
look,
looking-
here's a
you're
peer-reviewed
looking cut
publication
too, my
showing this
dude.
guy is wrong, showing that
All that walking in Vegas,
that
I
these
bet.
shots stay
All
in
that
the
expensive
muscle
ass water
and
in
the
Vegas,
local,
Jesus,
and
eh?
only go to local lymph node. And you know what? I went to that paper, Kevin. So this is 2.5 years later. I saw this, and I went to that paper, and you know what it was? It was a different vaccine, first of all, altogether. And get this, Kevin, Chris, and Steve. Do you know what tissues they looked at to look at the biodistribution? They looked in the muscle, a muscle biopsy, and the local lymph node. They didn't even look at any other tissues. So it's pretty easy to conclude it only goes to those two places if you only look in those two places.
There you go.
Right?
You only look if you.
And your listeners have to understand, Steve, because I honestly think that they're very confused, right? We hear all these experts on one side saying something. We hear all these experts on this side. Who the heck do I listen to? Well, you listen to the people who can explain the science just like Kevin did.
Absolutely. You know, and this reminds me that I first heard about what was going on with these vaccines because I was on a CCC call with Byram, and Byram was all excited that day, and he was saying, hey, I just got these results back from the Japanese government, and it shows that the shots don't remain in the arm like we thought. The biodistribution is huge. It goes to reproductive organs and all this, and this is kind of what really lit a fire under me. And once you told me that, then I called up Robert Malone and got Malone involved in this, and you know, things kicked off. We went on Brett Weinstein's Dark Horse podcast, and sort of, you know, the rest is history there. But it was Byram that was really the spark that really got me started when he discovered that information from the Japanese regulator that Pfizer had given to the Japanese regulator that he'd gotten under FOIA. But the other thing I want to mention is that it was Byram also that pointed out to me that, hey, we have contamination here, and then he pointed out this Michigan case where the contamination of the remdesivir, says that, hey, you're liable for that. And so it was Byram actually that got me to write that. Basically, that was the impetus for me writing the tweet saying, because I checked with the lawyers and they said, yeah, he's right, that he's correct, that this is, and I talked to Robert Malone, and he said, yeah, this is called adulteration. That's the term that you should use, and he agreed. And so this is what led to this tweet that now has, I don't know, 4 million, 5 million views. It'll probably be a lot more by the time most people are reading this. And so I want to credit Byram again for not only getting me started in this, but also as really he had the insight, because he knew about that Michigan case and he knew about the contamination, and he put sort of 2 and 2 together. And of course Kevin, you know, for his spectacular work in finding this, and always want to credit Chris, because he's just such a conveyor of truth and knows how to communicate these discoveries to people in a way that they can understand. I mean, all you guys are awesome, but I definitely wanted to make a call out to Byram, because he was really the impetus and inspiration for that tweet that I put out yesterday.
Well, thanks, Steve. But I told this David Speaker, who's you know my very close colleague, who's been working very closely with Kevin and to confirm what Kevin has been finding. And I said to this David the other day, and I said it about him and Kevin, that to me, so when to me the biggest wake up call prior was seeing that biodistribution, right? And a lot of people thought, you know, like, it wasn't like the biodistribution itself was novel to me. It was the fact that we were told, that I was publicly told, that wasn't happening, and then it was, and we were told there were proprietary aspects of these shots that couldn't be disclosed. So I thought, okay, one of those proprietary aspects is they've somehow found a way to tweak these lipid nanoparticles so they no longer get distributed through the body. But the reality is, I've been following this technology, and in the almost 2 decades leading up to it, they'd always been disseminated broadly. So it was that what was, to me, was remarkable was the obvious lying, and that was a big wake-up call. But to me, this is very different. To me, when I saw Kevin's data, and especially the work that he did with David, to me, this was the second big whoopsie moment, right? Whoops! This is a big whoopsie, and I say this is a bigger whoopsie because the other stuff they could write off in certain ways, but this is a huge whoopsie now, where we have a major World Health Regulatory Agency saying we have confirmed this whoopsie, and it is a whoopsie.
And we're doing
and you know, and to have that now, and this to me is the first time I believe, like we've had all these shocking revelations, but to me, this is a revelation that, in all right, it should be the final nail in the coffin, as far as I'm concerned, for these shots and getting a worldwide moratorium until the science can catch up. Because who can argue now? Like, that's why I point out, like, if you're going to start, like, you can call Kevin a misinformation spreader, you can call Chris that, you, Steve, and me, but now they have to call Health Canada. Like, Health Canada has basically said Kevin McKernan is absolutely correct, right? So they have to call them a misinformation spreader now, and they've been pushing these shots as hard as anybody,
right,
including your FDA and CDC. So, to me, this is
it's
huge.
Over.
Kevin has found the biggest whoopsie.
It's
that
over.
is going to be the hardest for all of the people pushing this narrative to shoot down. We've
Right,
got to push this forward.
Right, this has to end it. I mean, if anything is going to end it, I mean, this is the biggest nail in the coffin right now. This has to end it. Right? You guys agree? It's over?
I agree. I've told David Speaker again, and he gives all credit to Kevin as well, because Kevin was discoverer, and he's been working with Kevin on this. But in terms of David's context here in Canada now, I've nicknamed this work that he's just done with his preprint article and everything. It's the classic David versus Goliath. And again, his name's David Speaker, so that's where I've come up with that.
He's got diamond hands too, and he deserves plenty of credit, because you know, what we did is in a vacuum, and there's no reason for anyone to believe it until it reproduces. And in this circumstance, with the way peer review's behaving, where everything's getting rejected, this would have never got reproduced if people didn't step up to do it. And the people who step up to do this second get less credit usually, and just take just as much risk. So David's in the limelight for good reason, because He stepped into fire where he knew it was going to be. When I ran into this, it was an accident. I didn't know that I was stepping into this this horseshit, if you will. He willingly did, and it takes a little bit more courage than what I had to run into.
Chris, is this it? Is this the beginning of the end here? Are they gonna, is the FDA, what do you think's gonna, so let me ask you, what do you think is going to, so, and we should make this the last question because we've been here for close to 2 hours now, but here's the question, is what is your prediction for what is going to happen? What is the FDA going to do? What is the CDC going to do? What is the New York Times going to do? What is CNN going to do? You know, like, and what is mainstream medicine going to do? So the three things: mainstream media, mainstream medicine, and the mainstream medical authorities. Predictions. Kevin, we'll start with you. What do you think will happen?
I think media will ignore it. Medicine will quietly stop administering these. And who was the third one? The regulators?
Yeah, the regulators.
I think the regulators may take this opportunity as their escape hatch. Hey, you didn't tell us about this. It's time to throw Pfizer under the bus.
Yeah, throw Pfizer under the bus. And what about the lawmakers? That's the fourth one.
I think the lawyers are going to go crazy with this.
The lawmakers. No, Congress. People in Congress.
Oh.
Will they investigate? Will they call for hearings, or will they just ignore this?
I have a feeling there will be other distractions that continually emerge to take the attention in Congress off of this, whether it be Israel, Ukraine, or something else. I think they want to wash their hands of it.
Electing a speaker,
I don't know.
maybe electing a speaker, for example. Okay, Byram, four different.
Yeah,
four
so
questions.
I agree. I don't have anything to add to what Kevin said in terms of those four specific things. All that I will put on top of it is if these organizations that do not respond to this and do not take it seriously will lose a lot of public support and a lot of public respect, and deservedly so.
Absolutely correct. Chris, you think the FDA is gonna pull the plug on the vaccines? What do you think for regulators, Congress, media, and doctors?
They will ignore this as long as possible because that's what they've been doing. I believe, however, that there is real blood in the water this time, and for that I have to put on my market hat, looking at the stock prices of Moderna and Pfizer. There's some big money sneaking out the back door on those, and they usually know what's going on. So I think there's real trouble here. There's been some willful acts that have been committed. And you know, I don't take this from the data side. I love the data. Data is necessary, but insufficient. You have to win the mind share. So that's this common knowledge game again. So the prediction is that they'll ignore it, but when they don't, all of a sudden, it's going to be so fast. The media and politicians are in the same crowd for me. They are the least bold people on the planet. You know, Congress will grab the flag and run up the hill for like the last step. You know, after
Right.
after everybody else has already done the work, and then claim credit, right? So once the winds have shifted, I think watch out, because I do believe that there's a huge amount of pent-up energy because everybody knows somebody who's been injured or killed by these things at this point.
Yep.
That's devastating.
Yes. So I'll make a prediction. I'm going to guess that basically nothing's going to happen. They're going to ignore this, but we're going to continue to do the research. And as soon as we show that this stuff integrates into your DNA, the shit will hit the fan, and then it will be, then everybody will go and try to say, yes, let's get them, and everyone's going to go on the offensive against everybody else. The FDA will say, no, we didn't know, we were told by experts, da-da-da-da-da. Congress, we didn't know, we were told by experts. And everybody will shift over to supporting this, and they will put the blame on everybody else. What do you think?
Yeah. There is one concern I have with So I do think you had a tweet the other day, Steve, that no one's gonna investigate this. I actually think the NIH will fund something like this because they have $400 million of royalty on the line, and the conclusions will be written ahead of time. So we do have to be aware of that. That if the tide turns, who are we going to trust to do this work? It can't be the people who have conflicts.
No way. No way. No.
And if they do, and they try and publish that stuff,
That's really going to bite them in the butt.
Yeah, because I think they'll have great difficulty doing so and countering this story without making it obvious in their experimental designs.
Yeah, proximate origins is a perfect example of how they, continue to claim. In fact, my friend Sar Wolf, who's paid me $500,000 on the COVID vaccines, he's got a bet going with a guy who believes that who believes a natural origin story. So Sar says there's no way this is natural origin, and so they've got a $100,000 bet going. Let's do 200,000, 100 grand on each side on that one. But yeah, they've been keeping that going, and of course, that vaccines don't cause autism and all these other myths that have been going around for decades and decades. I mean, they're going to keep this thing going for as long as they can, but I think the shit's going to hit the fan pretty soon. That's my prediction.
Excellent. Well, I've got to get back.
Any last words? Okay. Any last words? So last words, Kevin, Byram, and Chris.
Give us, if anyone's interested in screening patients or interested in these PCR tools, contact me. You can find me on Twitter or on my Substack at the Pedalactone newsletter. And
what's the best way to contact you to get the kids?
Kevin.McKernan at medicinalgenomics.com or hit me up on Twitter, one of the two. And we'll eventually have them up on our website, but right now it's early stage, so we don't have it on a website, but there's been a backlog of demand. We just got a large lot in, so I think we can satisfy some of that, but we want to understand if there's other areas that need this, for, like the blood supply, like you guys brought up, it's an important point. If we need to screen that, then we have tools for it. So that's what I'm running out to do, is I got to go check on some of these lots that just came in, and I think that's the next step. We just got to see where is it, and if it's nowhere, then we move on, but we should fix the regulatory structure to allow this to happen, because it'll happen again. But if we find it, then we have a group of people that probably need to be sequenced very deeply to see if there's an integration event. Just finding the signal in PCR is step one. That it could be episomal, it may not be integrated, but we first need to screen patients that are of concern, and if they're positive on this PCR test, then those will be candidates for sequencing. But with that, I'll pass it off.
Thanks,
Yeah, for me, I'd say great seeing you, Chris and Kevin and Steve. Thanks for all your advocacy and making sure that honest, truthful science gets disseminated as far and wide as possible.
Thank you, Byron.
Likewise, and thanks to everybody on this call. You've all been tireless, perhaps none more so than Steve. Keep bulldogging this and just keeping at it when all other humans would have given up. So thanks for keeping on it. A lot of appreciation for what everybody brought here. So I'll do what I can to communicate it, and just great work, everybody.
Yep. Well, thank you all for being here and taking the time out of your day to chat with me about it and talk about these issues. It's super important. Thank you so much.
All right.
Bye, everybody.
Bye, everybody.
He's so based, man. Russ is so fucking based.
Honey of sweet truth. Sorry I was late, you lot. Guess who I'm talking to? Jordan Peterson. And we just did a fantastic 2-hour-long interview that if you are an awakened wonder and press the red button to become one, you can see straight away. Full 2-hour-long conversation with me and JP. Fantastic. And She Bites a Lot says, we want Gareth. Gareth is just there, and Gareth will return in peace times. When we have peace, Gareth will return. So that is why we've got to work together. When we hit 20,000 live viewers, Gareth will return. I'm seeing you now, there's nearly 11,000 when we get to 20,000. So if you're watching this anywhere other than Rumble, other than locals, I like to see you there in locals. I like you in my little community. I'm looking at the various screens where I can see you. Get over to Rumble. You might be watching this on YouTube now and you can watch it for a while, but a little bit later Dr. Bob Gill will be joining us. He used to just be a family doctor. Then the pandemic came around and he realized that he, like many physicians across the world, was being lied to. And you would have realized that, I suppose, maybe during the Sackler crisis, the opioid crisis, where they said, why don't you try a little bit of risk-free fentanyl? Try, if one's good, 20's fantastic. And those very same companies and that very same ideology, come the crisis time of the pandemic, who did we turn to? The just, the judicious, the awakened, the enlightened? No, the peripherally, perennially, and dreadfully frightened. That's who we turn to. Pfizer, sweet lady Pfizer herself, entrusted with undue power. Well, at least they're admitting, I can't even say it. You know what they're admitting, right? Let me know in the chat if you can see that. Can't say it while we're still on YouTube. And if you're an awakened wonder watching this on YouTube, if you got the notification, I suppose I'm grateful for even that, because we know whose tune YouTube dances to. The government say demonetize, YouTube says let's do it. Not over on Rumble, where people speak freely, where we can unify together. So Dr. Bob Gill's coming up a little bit later, and my apologies for my tardiness, because I loves you and I should never be late. Have you seen the breaking news? There's Trump's gagging order. We're talking about that. We're talking as well, of course, about Trump's former campaign lawyer, Jenna Ellis, pleaded guilty in Georgia. And what do you think about that, guys? Is that good? Is that good stuff? Or is this the final nail for Trump? Or will Trump, the perpetual phoenix, continue to rise? I think we've got that story here. And also we're going to be talking about Wikipedia and his funny joke, you know, Musk's funny joke. Did you see that? Wikipedia, that stuff. Love it. Oh, increase volume, please, it says here in the chat. So we've had stuff like that before, so I'm going to assume it's necessary. Get the volume up on the Rumble stream, please. Can someone tell me they've heard that and give me a response time for that? Thank you very much. Now, first of all, let's have a look at the Trump campaign lawyer, or at least a news report on that, over from our friends at the Legacy Media, right there.
Breaking news that just came through out of the state of Georgia, moving to a different criminal case, where Jenna Ellis, one of Trump's former attorneys, now joining Sidney Powell in pleading guilty. What do you read into that?
That's right. Well, you know, Willie, we've seen 4 pleas so far in that Fulton County case. 3 of them have come from people who behaved in a legal capacity. And I think going back to Michael Cohen, there's a lesson here that Trump didn't appear to have absorbed. Hell hath no fury, like a former lawyer whose own legal fees were unpaid and then is left to hang in the...
It's like the old saying goes, Hell hath no fury like a former Queen McSteve. Are you drunk, Russell? No, not for 20 years. For 20 years not drunk. That's not a catchphrase. That's never going to catch on, is it? Hell hath no fury like a lawyer not paid their fee. So I guess there's an indication. Still quiet, it says, Legi23. Louder. Someone's saying, turn the audio up louder. Make it louder, please, says Legi23. Can you tell me that you've got that and how long it will be, guys? Because it's super important to our audience. Lots of people asking for that on the chat. 13,000 people with us now. When we get to 20,000, we will parade Gareth around on roller skates. Okay, so let's see the rest of the story it's from MSNBC. Let's see what they're saying.
While that person is being investigated, that certainly seems to be what we're also seeing from Ken Chesebro's plea, from Sidney Powell's plea, and now Jenna Ellis's plea. All three of them are believed not to have had their legal fees paid by Trump, and then as defendants and or people under investigation, their own legal fees, in terms of their roles as defendants or investigated persons, weren't being paid. The poverty that caused for all three of these people could have been a major factor in their decisions to plea. Jenna Ellis...
That's no good, is it? If actually it's not about justice because they've not been paid their fees. So tell me, do you think this escalation spells the end for Trump? Put Y in the chat if you think this is the end for Trump, or N in the chat if you think that Trump will prevail. And if we want to get our facts straight, what better than a quick trip to Wikipedia, where the truth is available to you uneditorialized, except for in the view, of course, of Elon Musk, who says that he'll give it $1 billion if they change their name from Wikipedia to Dickipedia. I like this aspect of Elon Musk that's pure prankster, don't you? You know, that he just wants to use money. But last time he did something like that, he ended up owning Twitter, didn't he? Like, I'll just buy Twitter then. And then now he's got that bloody thing, that albatross around his neck, changed its name to X. Is this going to end up with Elon Musk owning Wikipedia with a bloody stupid name? Should he do that? Should it change it? Like the trickster in me rather likes the idea that Wikipedia will have to change its name sort of begrudgingly to Dickipedia and still take itself seriously, and that on their stationery it will say, oh, Dickipedia. It'll be irritating for them. We had Larry Sanger on the show recently. You familiar with Larry Sanger?
Thanks for being here.
He's one of the
founders
of Wikipedia, who sort of disavows it now. He says it's become editorialized. He says it's become a tool of the establishment. He says it's lost its way and lost its objectivity. Here's a conversation we had with Larry Sanger. He wants a kind of decentralized but unified approach to the internet. A lot of you, I think you're basically saying no when it comes to Trump, that this won't be the end of Trump, that is there anything they could say about Donald Trump that would disrupt your love of Trump, who you see as the berserker. We've got some great Trump stories coming up, so stay with us. The berserker in the chaotic, in the somewhat corrupted capital system. Okay, let's have a look at Larry Sanger now. You'll like this. I was talking to him about information, about how editorialized the data we consume is. Even something like Wikipedia, which I suppose is meant to be a neutral source of information, is heavily editorialized. But I had a little fool around with him as well. He's absolutely adorable.
I have to say, Wikipedia is not the only encyclopedia out there. And there is, I hope you're going to ask me that about the solution, because there is a very clear solution.
All right. Well, Larry, it would be quite remiss and almost unbearably recalcitrant for me now not to say, Larry, hold on, watch me do this because I'm a professional. Larry, this is very difficult for me to listen to, almost inducing despair. If only there was some sort of solution to this centralized, authoritarian, highly censored and cultivated space. Is there?
There is. I'm
that's
going
all
to...
we've got time for today. We're going to have to draw... That's a joke, Larry. That's a little... Matt, I'm part of the problem. Loved that. He loved that little joke. Okay, so
Thanks for being
you
here.
guys are, there's nothing
Appreciate it.
that could sway
Hope you
you
ain't got no beef
from
with,
your support
Dog
of
and
Donald
More
Trump.
Toad.
I'm looking at the
Video,
Rumble
I'm sorry,
chat
dude.
in particular. We're at 17,000 now. We are
Hope
so
you're doing well.
close to Gareth Roy being dragged around on a skateboard. Did we say skateboard or roller skates? He's going to be pulled out here. If that's what it takes, if that's what you want, if that's what's going to make you happy, we will provide it. For democracy and free speech lives on Rumble. If you're watching us on YouTube, we've got Dr. Bob Gill coming up, former family doctor turned radical crusader against the influence of Big Pharma. We'll be talking about a variety of subjects that YouTube would not permit. For now, though, Donald Trump is critiquing the Capitol. Not politically, though. I think from a real estate perspective. Let's have a look.
You know, big in real estate, I always kept clean properties. I like clean, well run. You know, tippy top, we say. Tippy
It's not a normal way to tippy top.
top. We want them to be tippy top. Well, our capital is the opposite of tippy top. It's a
Tippy top. That's what I think we're all saying. Whereas we want a bit more of a tippy top democracy, don't we? Or republic, you guys decide.
shit house. Horrible. It's horrible.
That's amazing. That is amazing. Why don't they understand that it is the personality that is powerful? That it is the power of charisma and personality that has raised him to a degree, to a point, to a plateau that